Hi everybody, it's Dr. Hall, and I'm sorry I can't be with you for this lecture, so here is a recording. It's not as good as live, I know, but so we're going to start talking about the nervous system now. So here we go. So as always, I want you to take a moment and stop and think about what are the functions of the nervous system. If we were in person, you would do a pair share on this, but just take a moment and think about what is your nervous system doing for you, right? Like, what are the things that it's doing for you? Okay. Now, you may have thought of it helps you feel things, right? You get sensations, right? If I feel the box here, right? Sensations. Oh, I'm also looking at my granola bar box, right? So it's bringing in things that are being received by my brain. It also is, of course, allowing me to move, right? So we've already talked about the muscular system, and so, of course, the nervous system and motor neurons are a really important part of that. You might have also thought about how your nervous system, particularly your brain, allows you to think, right? Or process things, process information. And so what it comes down to is we can kind of categorize all of the functions of the nervous system into three basic categories. So let's take a look. So the three basic functions of the nervous system is sensory input. So things, I guess it's going that way. So things that are coming into our brain from our periphery, from our eyes, from our ears, you're hearing my voice, right? From sensory receptors in our skin and in our muscles. Constantly we're getting input that is coming in from different parts of our nervous system and coming in to a central processing center where then it's being integrated. So you're hearing my voice and your brain is then making sense of it, right? You're processing that raw data and figuring out what are the words that I'm saying and what do they mean. You're looking at this diagram, you're seeing colors and lines, and then your integration system, which is your brain actually, is then figuring out, okay, well, what are those words that are printed? Oh, that's an eyeball, all those types of things. So we're integrating, and then we can send signals out from the nervous system to the body to tell it to do things. And whenever we're sending signals out away from the body, we call that motor output. Now, sometimes it's motor as in via a motor neuron and to a muscle, but sometimes it's to other things, right? So sometimes it might be to the tear glands in your eyes, right, to make tears, right? So it could be things other than actual skeletal muscles that are receiving that motor output. So this is that same information that I just talked about written down on a slide for you. So if you want to go back and review later, you can. All right, so those are the three functions of the nervous system: to take information in, to process it, and then to send information out to do things. And then let's talk about the actual physical components of the nervous system. So the first thing we're going to talk about is down at the microscopic level. So the nervous system is made of nervous tissue. I love that, right? That works really well. So we see here in the center of this histograph or this microscopic image, we see the neuron, kind of star-shaped cell, the big purple cell here, right? So this guy here is our neuron, and then we see all these little cells, these little purple guys, little dark purple guys around it, and those are the neuroglia. So the neuron is the cell that is receiving information and sending signals out, and the neuroglia are there just as support staff. They outnumber the neurons 10 to 1. Remember, the neuron is like our celebrity. It's like Lizzo or Taylor Swift, right? And then the neuroglia are the little support cells that are your PR person, your makeup person, your hair person, your personal trainer, your chef, your maid, all those types of things. So they're just there to support the neurons. And this is that information in text. So let's take a closer look at the anatomy of a neuron. We talked about this a little bit in the muscle physiology lecture, but we're going to go into a little bit more detail now. So you may remember this from high school biology, but so the main part of the neuron here is the cell body, often also called the soma. And then we have these little branchy things out here that receive signals that are called dendrites. We have this big, long, I like to think of it as a wire, which is the axon. And the axon is coated with a covering. And it is made by these little guys called Schwann cells. And so let's see, how can I demonstrate that to you? Okay. So let's imagine that my arm, let me back up, is a section of the axon. And then this yellow sheet of paper is a Schwann cell. And what they do is they wrap themselves around like a bracelet. I didn't plan this very well. They wrap themselves around the axon to form a covering of a substance called myelin. So we have these little cells all up and down the axons, not all axons, but most of them, that give it this covering, which is called the myelin sheath. So each individual Schwann cell then is contributing to this covering around the axon, which is called the myelin sheath. There are these little spaces in between those Schwann cells, and those are called the nodes of Ranvier, which is a very romantic sounding name, which I like. And then, of course, at the end of the axon, remember these axon terminals that look like little frog toes, and that's where we're going to then send a signal on to the postsynaptic or receiving cell, which might be a neuron, might be a muscle cell, could be a lot of different things. Okay. So here are a few more slides, again, just going over that detail so you can kind of look back at these for review. So our dendrites, which receive signals, those branches, the cell body or soma, it's like the main part of the cell. You can also receive signals directly to the cell body. The axon itself, right, which is this long extension of the cell that then sends signals out. So I like to think of it like a wire. And that myelin sheath that I talked about that's formed by the Schwann cells that wrap themselves around the axon acts kind of like an insulator for a wire. So just like, you know, when you buy a wire from the hardware store, it has that plastic coating on. That insulator helps it conduct electricity better. The Schwann cells in the myelin sheath work similarly, not exactly the same, but similarly for our neurons, and we'll talk about that a little bit more in the lecture. Then, of course, again, the axon terminals, meaning the ends of the axon, these little knobby things where a whole bunch of fun stuff is going to happen, as we talked about previously with the neuromuscular junction. So far we've talked about the functions of the nervous system, sensory input, integration, and motor output. We've talked about the microscopic composition of the nervous system with the nervous tissue and then the parts of the neuron. And now we're going to talk about how we kind of organize and think about the organs that are involved in the nervous system, because of course the nervous system is composed of several different organs all working together. So we divide these organs into the central nervous system and the peripheral nervous system. So central means it's kind of central, right? It's like headquarters. And then peripheral means like out to the side. So that works pretty well. So for central nervous system, we have two main components shown here in purple. So we have the brain and then the spinal cord. So that is the central nervous system. And I'm going to erase those. And then for the peripheral nervous system, that is composed of all these nerves that we see out here. Okay, it's also got some ganglia in it, and you're like, what's that? We'll talk about that later, but it's mostly nerves and some ganglia. So central is the purple stuff. That's headquarters where a lot of interesting stuff is happening. Peripheral is the nerves. So central is brain and spinal cord. And here is that information. Now, the other thing that I didn't say yet is that the central nervous system is where our integration happens. So that's where all this kind of processing and figuring things out and comparing it with other information that's coming in or going out happens. And the peripheral nervous system is really just actually sending the information. So if it is coming from the body into the central nervous system, that is sensory. And the other word that we give sensory input is afferent signals. afferent with an AFF. And the way I remember that is affection. So you have affection towards, you want to get you're affectionate to, you go towards the thing. So we're coming toward the CNS, it's afferent. And then if it's going from the central nervous system out to the body, that is motor or efferent. And I think E exit, we're leaving the central nervous system and going out to the periphery. So those two new vocabulary words, afferent coming into the central nervous system, efferent going out from the central nervous system. So what are nerves anyway? So we have nerves that are, where are they? So they go throughout the entire body. So they come out from the brain and they come out from the spinal cord, and then they travel to all the different parts of our body where we need to send motor signals or receive sensory information from, which is basically every part of our body. So we have nerves that travel all over. They often travel alongside or in parallel with our blood vessels because, like, well, we've already got somebody traveling through there. We might as well bundle them all together. So they often travel with blood vessels, but not always. And yeah, they range a lot in size. Some of them are super big, like as thick as a watch strap. And some of them are really just tiny little microscopic nerves. So we have all different kinds of nerves that branch out and provide a communication system to all parts of our body. So again, this is just that information. And so I'm going to show you a picture. And this picture is from a cadaver, not one of ours because we don't have permission to use those photos, but from a different one looking at the sciatic nerve. Now the sciatic nerve is a big nerve. It's the biggest one in the human body, and it's located in your buttock. It's underneath the gluteus maximus muscle, which you'll be learning about soon. And so I'm going to show you a picture of somebody holding a sciatic nerve. If you're squeamish or you don't like the idea of looking at a picture of a cadaver, I'm going to have you go ahead and close your eyes now. So here is the picture, and I think you can see quite clearly, right? This is the sciatic nerve. It is thick, right? It is really big, okay? So now we have to think about, well, why is it so big? Like, what makes one nerve bigger than another? What are they actually composed of other than nervous tissue? You know, it's nervous tissue, but like, what components of nervous tissue are actually found in a nerve? So those of you with your eyes closed, keep them closed until now. Okay, so it's safe to look now. So nerves are actually bundles of axons. It's like, okay, we have all these axons that are sending signals out to the body or sending signals back towards the central nervous system. So we're going to bundle them all together, and we do that with a bunch of connective tissue, and then that's what a nerve is. So interestingly, there are no neuron cell bodies in a nerve at all. All of the neuron cell bodies are located either in the central nervous system, so your brain and your spinal cord, or in those ganglia that I teased you about, which are close to the spinal cord. So what's really interesting, for example, is that sciatic nerve is carrying axons from neuron cell bodies that are in my spinal cord in my lower back. And then those axons are like, some of them are 3 feet long because they go all the way down to my toes. So the neuron cell body is all the way up in my lower back, but yet that axon is going to travel through the sciatic nerve and then down further to my legs. So it's just pretty amazing when you think about it, right? They can be like 3 feet long, these axons. Okay, but no neuron cell bodies live in the nerve. A nerve is just a bundle of axons. It's like, here's a bunch of cables, here's a bunch of wires. We're just going to wrap them up together so they can travel as a unit. That's what a nerve is. It's a bundle of axons. Here is that same information. Now, it's important to realize that they're going to have both types of axons traveling through them most of the time. Not always, but most of the time. So let's talk about what that means. Oh, my head is in the way, but that's okay. All right. So I want you to imagine that this thing down here is a nerve. And so a nerve is just a bundle of axons. And so if I look inside that nerve, I will see two different types of axons. I will see motor or efferent axons, which are carrying signals from the central nervous system out to the body. And I will also find sensory or afferent axons, which are taking signals from the body to the central nervous system. So it's interesting, every single axon is a one-way road. You're either a motor or efferent axon, and I'm going to use those terms interchangeably. They both mean the same thing. Or you're a sensory or an afferent axon. It's like you can't be both eastbound and westbound 94 at the same time. You're just a one-way axon, okay? All right. Here is that text information. So again, the axon is either sending signals from the central nervous system to the body, in which case it's motor or efferent, or from the body to the central nervous system, in which case it is sensory or afferent. So let's come back then to how that signaling actually happens. How do we send those signals? So we're going to talk about neuron signaling. We already talked about this a little bit when we did neuromuscular junction. So these are the steps that we're going to go through for neuron signaling. So we start with a resting potential, then we're going to reach threshold, then we're going to send an action potential, then release neurotransmitters, then those neurotransmitters are going to be caught by receptors, and then those neurotransmitters are somehow going to come to an end. And there are two ways to do that, through degradation or reuptake. And so the next bit of slides goes into detail on all of those things. So here we go. So remember, we first need to establish a charge gradient, right? This resting potential where we have a lot of sodium on the outside of the cell and not a lot of sodium on the inside of the cell, which makes the outside of the cell relatively positive, right? And we need to do this because that movement of charge signals is what an action potential is. So in order to do that, we have to set up this resting potential. So at rest, the neuron uses ATP to pump sodium outside of the cell so that the outside of the cell becomes relatively positively charged compared to the inside of the cell, which is then relatively negatively charged. And you can see here, if you took a little mini voltmeter, it's a difference of 70 millivolts, which is like, that's measurable, right? That's kind of interesting, right? So we actually have this charge gradient when the neuron is at rest. Same thing with with a muscle cell, right? So across the sarcolemma, we have a resting potential as well. But so the neuron is going to have a resting potential set up across the outside versus the inside of that axon. Here is the text information for that. So this is our preparation step. While the neuron is at rest, we establish the resting potential so we have the ability to send the signal when we want to. Next comes threshold. So a threshold is like a crossing. It's that little, it's this little thing, right? Like on the floor in the doorway. And it comes from a long time ago in like medieval Europe when like literally people just peed on the floor and stuff. I mean, it was really strange and really weird. And so they just had like hay and straw on the floor, and they called it the threshings. And then so it didn't all go outside the room. They had this raised little lintel at the doorway to keep all the threshings in the room. So it was called the threshold, which is really weird to think about, that people just like, yeah, it's kind of just like living in a barn. But anyway, so the threshold, right? So this image is sort of like, this is, you make the decision to go forth, right? So something causes a neuron to reach threshold. Usually it's that it's received a whole bunch of other signals that then bring it to a state where it's like, okay, now I'm going to send my signal. So for one reason or another, a neuron reaches threshold and makes the decision that it's going to send a signal. Okay, so once it's received enough stimuli, it's said to reach threshold, and now we're like, okay, let's do this, and we're going to send an action potential. So you may remember that to send an action potential, what happens is we open sodium gates in the plasma membrane of the cell. So what you can see happening here is we had lots of sodium ions outside the cell at rest, and then once this gate opens up, they rush down in. Okay, this is called depolarization, right? We were polarized with all the positives outside and all the negatives inside. But when we open the gate, right, They all rush in, and then we lose that polarization. So that's why they call it depolarization. So what you can see on the right side is the voltage then. So movement of charged particles is electricity. We can measure that as voltage. So we have this little electric signal then that is caused by that movement of charged particles. In this case, positively charged particles, sodium ions. So that action potential then is going to sweep down like dominoes the rest of the axon from the beginning of the axon all the way down to the end. The beginning of the axon is this little part where it attaches to the neuron cell body here called the axon hillock. It's kind of like a little swollen part of the axon, and somebody thought, oh, it looks like a little tiny hill, so I'll call it a hillock. Okay, so the axon hillock is where it starts. So it starts at the very beginning of the axon. Those sodium channels open, and then boom, 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 boom. It's going to go down the whole rest of the axon. And remember, this is an all or nothing event. You either send the action potential or you don't. There's no such thing as a stronger or a weaker action potential. You send it or you don't. And so what we can see here is that we've just opened these gates and sodium has rushed inside. Over here, right, they've already been there, done that, and they've reestablished for the next time. And these guys over here, they haven't rushed in yet, right? So right now the action potential is going down this way. Okay. Here is that same information just written out. And action potentials are ridiculously fast. So this is a cool little video where you can see them actually measuring the speed of the electrical signal from one part of a nerve to another part of a nerve, and nerves are just bundles of axons, right? So that's telling you how quickly it's traveling down an axon. And it moves at about 50 to 150 meters per second, which is over 120 miles per hour. So it could travel down the entire football field in 1 second. And I don't think any of our football players can run that fast, right? So super duper fast, which means it only takes 3 to 5 milliseconds, which is 3 to 5 1,000ths of a second, right, for it to get down the length of the axon. Or you could think of it as we can send 200 to 300 signals per second down an axon. It's super, super duper fast. Maybe really, really fast. Now, how does it get so fast? Aha! This is why we have the myelin sheath. So remember, those Schwann cells that wrap around the axon and create this, it's kind of this fatty substance, this myelin that they're filled with. So it makes a really good insulator. And what happens then is that the action potential doesn't, let's see, do I have my little pen? Where's my cursor? Okay. The action potential doesn't go just do chug, 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 chug down the axon. Instead, it goes bleep, 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 bleep. I'm sure it makes that noise, right? So it just jumps, it skips. So instead of just being like boom, 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 it goes bing, 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 and it goes down the axon, and it's super duper fast, which is really cool. So we love our Schwann cells that are filled with myelin. We love this myelin sheath because it's what allows our axons to send signals so quickly. And when you're a really long axon, like the one that's going all the way to or from my toe, thank goodness. Otherwise, like the signal just wouldn't even ever get through before I died. Okay, so it just wouldn't happen. So again, this is that same information. And myelin is this kind of fatty substance, so it looks kind of glistening off-white, kind of slate yellow in color, kind of an ivory color. So for those who want to see it, I'm going to show you an image not from a cadaver, because colors are different in cadavers because they're not alive anymore. We're going to look at an image from a surgery on the nerve in somebody's calf, actually. So if you don't want to see it, you're going to close your eyes in 1, 2, 3. And so here we see they have opened up somebody's posterior leg. They've opened up somebody's calf. And here, I don't know if you can see, probably red isn't the best color. Let me do blue, right? So here you can see that is the nerve. That is a tibial nerve. It's the nerve in your posterior leg. And you can see that kind of ivory or pale yellow kind of glistening color to it. You can also see here if you're interested. So here's part of your gastric medius muscle. They've kind of pulled that to the side to see the nerve. And then there's some adipose tissue, right, in your hypodermis underneath the skin. All right, it is safe to look for those of you who didn't want to see that image. So of course, when we reach the end of the axon, then we get to that synaptic cleft, that synapse, that gap that is between the axon terminal and whatever the receiving cell is. So the receiving cell is usually called the postsynaptic cell because it's the cell that's on the other side of the synapse. It's after the synapse. So the postsynaptic cell, which may be another neuron, or it could be a muscle cell, right? So we have this gap, and so if anybody's ever been to London and ridden the underground, which is what they call the subway there, there's a little gap between the platform and the trains, and they have all these little signs that say mind the gap, right? Be careful, don't twist your ankle, don't get your foot stuck in it. So there's mind the gap, there's a little space there, and so you'll remember, of course, that we need a special way to get across that space because our action potential cannot cross that space. This is the text information. So to cross that space, we're going to convert that electrical signal of the action potential into a chemical signal of a neurotransmitter. So we are going to release a neurotransmitter via exocytosis from the axon terminal, and it's going to drift across the synapse and land on the postsynaptic cell. So these neurotransmitters are chemicals that act as signaling molecules then, and they can cross the synaptic cleft. We've learned about one of them so far, but there are many different types of neurotransmitters that our bodies use. So we learned about acetylcholine, which is a neurotransmitter that's always used when a motor neuron is signaling a muscle cell. But other neurotransmitters that you've perhaps heard about would include serotonin, epinephrine, norepinephrine, which are often known as adrenaline and noradrenaline, dopamine, GABA, and many others. Okay, so there's lots of different ones. They do different things. We've already learned about acetylcholine, which is in the neuromuscular junction. So let's also remind ourselves how neurotransmitters get released. How does the action potential trigger the release of those vesicles, those synaptic vesicles? Well, it's going to be calcium again, right? So let's just refresh our memory on why calcium is so important, right? So it's important for bone strength. It's that component of the extracellular matrix. It's part of the ground substance that makes it hard. It is important for muscle contraction because the sarcoplasmic reticulum has to release calcium in order for troponin to move tropomyosin off of the binding site so myosin can form a cross bridge, do power stroke, release, reset. And it's important for nerve signaling because when that action potential reaches the axon terminal, it is the opening of a calcium channel that allows calcium to move into the axon terminal that then triggers the neurotransmitter to be released. So let's take a look here. Let me switch back to red. So my action potential is coming down the axon. I reach the axon terminal. That causes this calcium channel to open. Calcium then, let's use green. Calcium then comes into the cell, and that causes these synaptic vesicles that are containing neurotransmitter to then, let's switch to purple. Where'd I go? I don't know. Okay, here we go. Then do their exocytosis and release neurotransmitter into the synapse. Calcium, man. No wonder why we have to control its homeostasis with calcitonin and parathyroid hormone. Okay. This is just that information in text. Here is just another diagram of the same thing with that excellent animation of exocytosis, releasing the neurotransmitter into the synapse. One more time. Here we go. Awesome. Okay. And this is all that information in text with my head in the way of the animation. So after the neurotransmitter gets released into the synapse, it has to be caught by the postsynaptic cell, whether that's another neuron or a muscle cell or something else. And so that cell, that postsynaptic cell, has to have receptors. And those receptors are going to be specific for that neurotransmitter. So acetylcholine will only fit and be able to be caught by acetylcholine receptors. Serotonin will only fit in and be able to be caught by serotonin receptors, and on and on. So they're very specific to that neurotransmitter molecule. Now, the other thing that's interesting about receptors is we saw in the neuromuscular junction that when acetylcholine bound to the receptors on the sarcolemma, on the muscle cell, it caused then that muscle cell to send an action potential down its sarcolemma. So that was excitatory. It told the muscle cell to do something. Now, sometimes neurotransmitters tell cells, shh, don't do anything. And that's called inhibitory. So some types of neurotransmitters are excitatory and some are inhibitory. So some are telling cells, yes, do the thing, excitatory, and some are telling cells, no, no, don't do the thing, inhibitory, which is kind of cool. This is that same information. Okay, we have to end the signal. So, because if we just let the neurotransmitters stay in the receptor forever and ever and ever, then the cell is going to keep either doing if it was excitatory or not doing if it was inhibitory, whatever it was. So we need the ability to turn that signal off. So we have to get that neurotransmitter out of there. We have two ways of doing it. One is enzymatic degradation, using an enzyme to degrade or chew up the neurotransmitter, and the other is reuptake. So we've already seen enzymatic degradation because that is what we saw in the neuromuscular junction. So you might remember when I threw the paper airplane, which was acetylcholine, and it was caught by the receptor, not very well because it didn't make a good paper airplane. But anyway, once it got there, then an enzyme came in, and the enzyme is called acetylcholinesterase with the ACE on it. That tells you it's going to chew up acetylcholine, and it rips up the acetylcholine, as you can see. So that enzyme now, it chews up the acetylcholine into little bits, and then those little bits get recycled, and then the cell will build acetylcholine out of it again. So as soon as acetylcholine is released into the synapse, some of it's going to bind right away, and then like acetylcholinesterase, the enzyme is just there and ready. It is ready to just chew it up, okay? And then it's going to be gone. Then it can't send the signal anymore. So that's enzymatic degradation. The other way that neurotransmitters get taken out of the synapse so that the signal ends is something called reuptake. So here, let's see what color is going to show up. Well, maybe bright blue. Let's see. So here we see my exocytosis of my synaptic vesicle releasing this neurotransmitter into the synapse. And the neurotransmitter binds to the receptor, but then look, it goes, and we pump it back into the axon terminal. So this is called reuptake, because the axon terminal is like, here you go, here's a neurotransmitter, and then it's like, oh, no, taking it back. So it just allows the neurotransmitter to be there for a little bit, and then the axon terminal kind of pulls it back quickly thereafter. So instead of Cookie Monster chewing up the cookie, right, it's like somebody giving you a cookie, and then after a moment they take it right away from you. So this was adorable. So let's just have to watch this. He's eating a cookie, and then reuptake, taking it right back. Poor little kid. I just love how then he looks around. He's like, I am not sure what happened here. Let's watch it one more time because it's so adorable. He's eating a cookie. The neurotransmitters in the synapse. Oh no, took it right back into the axon terminal. So that is reuptake. Okay, so reuptake is kind of like recycling. We're taking that neurotransmitter right back. And so serotonin is a neurotransmitter for which we do reuptake. That's how we end a serotonin signal in a synapse, is we do reuptake. We pull it back into the axon terminal, takes backsies. So what we're going to do now, the rest of the lecture, is we're going to do some clinical cases. Some of these are based on patients I actually saw when I was practicing. Some of them are not based on patients I saw, but on other things that happen to people. And hopefully it'll be useful for illustrating some of the concepts we've just reviewed by talking about when sometimes they're not working correctly. So first case is Ned's nerve. Ned sometimes has trouble staying awake in class. I know none of you can relate. And let's take a look at Ned's position here. And you notice that his arm is probably getting really compressed against the desk there. So that's going to be relevant in this situation. So one day he wakes up at the end of the class and he can't feel parts of his hand, and he also can't move it normally either. So it's numb and it's kind of a little bit paralyzed. So he's like, like, what is going on? He goes to the health center and they're like, dude, you irritated your radial nerve. Well, that's clear as mud. So let's talk about it. Right? What's a nerve and why did this happen? So the radial nerve, nerves are bundles of axons, right? The radial nerve is located in your arm. Where's my little pen? And it starts way up here in your upper arm, and it travels really close to your upper arm bone, which you now all know is the humerus, travels behind it, and then comes around to kind of the back of your hand. So I'm too far away for, okay. So it comes from down under here, and then it spirals around, and it comes up there. And that nerve is really vulnerable to getting compressed when you like fall asleep with your arm on something that's really hard and unyielding. And so that nerve also is not only vulnerable to getting compressed, but it is carrying both motor fibers that are coming from your brain and spinal cord out to the arm that would normally help you move your muscles, and it's carrying sensory or afferent axons or fibers, which are taking sensory information from your hand, wrist, forearm, and arm up into your central nervous system. So what happened here? So Ned's nerve got compressed and it crushed the fibers, and each fiber is an axon. So we can see this compressed nerve down here, and you can imagine that if the axons have just been squished, they're not going to be good at sending action potentials. They're like, dude, like, we are injured, we cannot send signals right now. So if you can't send motor signals, efferent signals from the CNS to that part of the body, then you're going to be paralyzed. You're not going to be able to move because the wiring is not working. And if the sensory axons are damaged and not able to function, then you can't receive any sensory information from that body part. So you're going to have that be numbness. Right? So nerve compression like this is pretty common. Anytime you've had like your leg fall asleep or something, that's what this is. Now, usually it's the sensory or the afferent axons that are the most vulnerable to these. So you get that numbness. And then when you get that pins and needles, what's happening with pins and needles is all of a sudden like your axons are trying to reestablish their resting potential, and they send all these action potentials like as they're trying to get better. It's just the worst. Okay, so the good thing is that relatively mild compression usually heals on its own. Sometimes it takes a while though, sometimes even a couple of days if it's pretty significant. All right, so the compression of his nerve from his sleeping position caused some minor damage to the nerve. So those axons couldn't send their signals. With no motor or efferent signals, you have no motor movement, you have paralysis. With no sensory signals, you have numbness, right? And again, this will heal in time as long as it's not incredibly severe. Next case, Nora's numbness. So Nora cuts her finger and she needs stitches. So she goes to Student Health Center, and you can do this and they'll do stitches for you and it's free, which is pretty awesome. And when she's there, the nurse practitioner injects a local anesthetic called lidocaine, and it goes numb, right? So if you've ever had numbing medicine, it's probably been lidocaine or Novocaine or one of the other canes. They're all related chemicals. And Nora's like, well, how does that actually work? And so the nurse practitioner says, it stabilizes the neuron's plasma membrane and prevents depolarization. Nora's like, huh? Okay, let's take a look at how lidocaine works. It's super cool. All right, so it stabilizes the plasma membrane and prevents depolarization. And it does that by blocking the sodium channel from opening. So I forgot to put an animation here, so I didn't want you to see both of these at the same time, but oh well. Okay, so this upper one is without the lidocaine. So this is normal when you don't have numbing medicine, and you can open your sodium gates and sodium can rush in and you can send an action potential down the axon. With lidocaine, however, lidocaine sits in those sodium channels and blocks them. So sodium can't rush into the axon. And so sodium ions can't rush into the axon. You can't have a movement of charged particles. You have no action potential. So it's totally cool. So like, even if the neuron is reaching threshold and it's like, dude, I need to tell the brain that somebody is poking a needle in our finger to give us stitches, lidocaine is like, right? So that's really cool, right? It's super cool. And just so that you know that I am not the nerdiest person, you know. Well, no, I am, but I'm not alone in my nerdiness. This is from a friend of mine that I went to high school with, and she is a nurse practitioner. And at the end of their nervous system unit when she was in school, she baked everybody a cake. And this cake is showing a really cool thing. So it is showing the plasma membrane. I'll use purple. This is the plasma membrane of the axon. This is a sodium channel that is letting, never mind. This is a sodium channel that is letting sodium ions in for an action potential. This is the action potential. And then this yellow is lidocaine blocking the gates and preventing that signal from being sent. Okay. That was a little bit too much time for my friend's cake. Okay. All right. So the nice thing about lidocaine is that it's temporary. So this effect wears off. Your body is able to eventually get it out of those sodium gates so that then you can send signals again. And so your numbness will wear off in a couple of hours generally. Okay, so that is how lidocaine works. It blocks those sodium channels, so then you can't send an action potential down the axon. Next case. So Sarah is really interested in she's wondering about how sarin, which is a nerve gas, works. And she's read that it's an acetylcholinesterase inhibitor, and so she asks what that means. And so this picture is, I have a little bit of qualms about it, but hopefully that man got resuscitated. But so in 1995, there was a sarin gas attack, a terrorist attack in the Tokyo subway. Actually, somebody released sarin gas, which is a nerve gas, it's a poison gas, and many people actually died. And so this is a picture from newspapers at the time. So how does sarin kill people? So it's an acetylcholinesterase inhibitor. If you're inhibiting something, that means that it can't work. So sarin blocks the enzyme, the acetylcholinesterase enzyme that normally breaks apart acetylcholine, right? So if you can't break apart acetylcholine, then acetylcholine is always sitting there in the receptor and always sending its signal to the postsynaptic cell. And in much of the body, that postsynaptic cell is a muscle cell, right? So that we use acetylcholine in the neuromuscular junction between the motor neuron and the muscle cell. So that is actually how it kills people, because you can't relax your muscles. If acetylcholine is always sitting there in the receptor, then the muscle is continually sending an action potential down its sarcolemma, down the T-tubule, sarcoplasmic reticulum is releasing calcium, like you're doing the whole thing, you're doing muscle contraction constantly. So people who've been exposed to sarin nerve gas, much like people who have organophosphate pesticide poisoning, which does the same thing, it blocks or inhibits acetylcholinesterase. So the acetylcholine is sitting there, the muscles are constantly contracting, right? So they'll often have like a grimace or weird, like their limbs will be contorted in weird shapes because like all their muscles are contracting at the same time. And what kills you is that your diaphragm is a skeletal muscle that relies on acetylcholine. And so your diaphragm is constantly contracted. And so you can't then, you're constantly in the inhale position. You can't exhale and then take a new breath. So what happens with sarin nerve gas or organophosphate poisoning is you suffocate, basically, because your diaphragm doesn't work. So first aid to somebody then would be A, make sure you protect yourself, and then B, give them ventilator support. So bag valve mask for the EMTs in the group, and then put them on a ventilator. Yeah. Okay, next case. Mia's myelin. So Mia all of a sudden starts having trouble with her vision. And it turns out she has an unusual cause of vision trouble, which is MS or multiple sclerosis, which often affects the optic nerves, which are the nerves to our eyes. So it turns out that MS is a disease where parts of the nerves in and other sections of the brain or spinal cord lose their myelin. So remember that myelin sheath is what allowed axons to send their signals rapidly through that saltatory conduction, just skipping from node of Ranvier to node of Ranvier. So what happens then is that the signal is so slow that it just doesn't even get there, right? Or it would take years to get there, so then it just doesn't work. So depending on what parts of the nervous system are affected by MS, you can have a whole variety of symptoms. You can have sensory symptoms like vision problems. You can have motor symptoms like paralysis or weakness. You can also have integration problems like thinking and mood and fatigue. So MS is a really fascinating and devastating condition, actually, because we have some treatments for it, but we really aren't good at treating that. Unfortunately, I have a friend who's pretty badly affected. So that is MS, disease of the myelin. Next, Sally's serotonin. So Sally has been having trouble with symptoms of depression, and her doctor says that people with low depression often tend to have low brain levels of serotonin. And this is a model of our understanding of depression, which has since been called into question, but it's still used widely as kind of a theory of how depression works. But anyway, they prescribe fluoxetine, which is Prozac, an antidepressant for her. Now, fluoxetine is in a class of medicines called SSRIs, and that stands for selective, meaning it's choosy, serotonin, meaning it's selecting for serotonin and serotonin only, so it's specific to serotonin, reuptake inhibitor. Haha, so now you know what that does. Okay, so remember, serotonin gets taken out of the synapse and its signal ended by the process of reuptake. So here we have our serotonin neurotransmitters, right, in our vesicle being released into the synapse, and then they're not there very long before they get reuptaked. That's not a word. They get taken right back up into the axon terminal, which ends the signal. So this kind of a hypothesis of depression is that you don't have enough serotonin. So we would like the serotonin that you have to be able to stay in the synapse a little bit longer so you can get more use out of it. So what reuptake inhibitors do is they actually block this reuptake process, and therefore the serotonin hangs around in the synapse longer and has more opportunity to do its thing. So you might have fewer molecules of serotonin, but they get more time to send their signal. So that is kind of the theory behind why these medications work. There are some problems with that theory, but the medicines actually do work. We just aren't necessarily that great at understanding exactly why, but the medicines do actually work for a lot of people quite well. All right, so neurotransmitters and mood. So this is a real oversimplification, but it's just one of those really interesting things that students like to hear about. So when we use all these different neurotransmitters in our brain, right? In our brain, we haven't talked about the brain yet, but that's where our mood and our thinking, which is cognition, that's where all that is happening, right? Not out in the nerves. There's not even any neuron cell bodies there, right? The important stuff is happening in HQ, right? In the central nervous system. So very broadly, and again, it's an overgeneralization, but norepinephrine tends to kind of rev us up and make us alert and help us concentrate and give us energy. Whereas serotonin can be important for memory, and if you have problems with it, sometimes obsessions and compulsions like obsessive compulsive disorder, OCD, and we see sometimes problems with anxiety or impulse control or irritability. Sometimes there's something going on with serotonin or norepinephrine. Dopamine is our pleasure or reward chemical in our brains, and it is what is released in massive amounts if you take heroin. So please don't. But so then both dopamine and norepinephrine seem important for attention and maintaining attention, and both dopamine and serotonin are important for appetite and sex drive and sex behavior, as well as aggression. So you can also see too how some of the side effects that we get from certain types of antidepressants, like a serotonin reuptake inhibitor, one of the side effects can be your appetite can be off or your sex drive can be off. So anytime we monkey with one of these neurotransmitters, right, we can get side effects depending on what types of behaviors or things that particular neurotransmitter is involved in. So kind of neat to think about. So in summary today, we talked about how the nervous system has input, which is sensory or afferent. Then we have processing and integration, which happens in the central nervous system. And then we have motor or efferent, which is our output, going from the central nervous system out to the body via the peripheral nervous system, right? So our peripheral nervous system are our nerves, our wires, basically, allowing the central nervous system to talk to the body and vice versa. The process of neuron signaling, right? So we start with that resting potential. We're going to use ATP to pump sodium outside of the cell to establish that resting potential with high concentrations of sodium ions that are positively charged outside the cell. And then once we reach threshold, we decide we're going to send an action potential. We open those sodium gates, we get depolarization happening, and then that action potential is going to travel boom, 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 via saltatory conduction down from nodeuron VA to nodeuron VA, thanks to the Schwann cells in the myelin sheath. When we reach the axon terminal, then those calcium gates are going to open, which causes the synaptic vesicles to release the neurotransmitter into the synapse. And then that neurotransmitter crosses the synapse and binds to specific receptors, which will either excite or inhibit the receiving or postsynaptic cell. And then we're going to end that signal from the neurotransmitters either with an enzyme, like acetylcholinesterase, enzymatic degradation, or reuptake like we do with serotonin. And that's it for the intro to the nervous system. Have a great weekend, everybody.